Hi, my name is Lauren Kirkland, and today I'm here to talk about my final lab project in my Microbiology 214 class. And we were assigned a lab that was called Save the Patient. And in this lab, we were each assigned a specific unknown organism. And it was our responsibility through a number of tests to see if we could identify what that unknown organism was. So in short, this video is going to recap the purpose for this, the methodology behind the test that I did, the results, and then a short discussion. So first, we're going to talk about the purpose of this experiment. Not only was this an assignment, but this ties into a lot of purposeful things for me. As a nursing student, hopefully a future nurse, this knowledge gained and a greater insight will allow me to be more prepared in my workplace and then even in upcoming classes. I just have a better understanding of bacteria and microbiology and what tests to perform and different reactions and so forth. So the why behind this experiment was really special to me and important to me, and really applies to what I have lying ahead. So now we're going to talk about the different tests that were done that I did. I did 4 tests. On the first day, I did a gram stain and a catalase test. A gram stain test determines the reaction of our bacteria. And this test has 4 different reagents. You have your crystal violet, your iodine, your decolorizer, and the safraran. And I'm going to tell you a little bit about each one of those, okay? So to perform the gram stain test, what you're doing is you have a culture and you're putting this stain or dye, each one of these, on this slide to eventually look at this slide under a microscope to see what type of bacteria you have. So the first one that you will put is the crystal violet. And when you put the crystal violet dye on your slide on the culture, it will turn purple. Then you will apply your iodine, and this iodine breaks away at the plasma membrane. Then you have your decolorizer, which oftentimes is alcohol. And what this does for gram-positive bacteria, it is still purple. And for gram-negative bacteria, it strips that color away. And then you have your safraran, and when you add this, if you have a gram-positive bacteria, it will still remain purple. And if you have a gram-negative, then it'll be more pink or red. So these results tell us if our bacteria is positive or negative. And this allows a lot of insight of what test to do next. So then, the catalase test. The catalase test, the purpose of the catalase test is to see if the microbe has an enzyme called catalase that is capable of destroying hydrogen peroxide. And if it does destroy hydrogen peroxide, it forms into water and oxygen. And the reagent used in this test is hydrogen peroxide. The results for this, or how this is performed, is you have an inoculum from a pure culture. You let this grow overnight, and then when you're ready to do this test, you pull it out of the incubator, and then you'll smear some on a microscope slide, and then you will put a drop of hydrogen peroxide on this culture. If you see copious amounts of bubbles, then you know it's catalase positive, and it does contain that enzyme. If you don't see any bubbles, it is negative. The next test is the blood hemolysis test. The blood hemolysis test and the mannitol salt agar test I did on day 2. The blood hemolysis test, the purpose of this is it provides information on what hemolytic enzymes the bacteria possesses, whether or not the the bacteria can destroy cells or if it can digest that hemoglobin inside. There's no reagents used, and the results in this, you're either going to have an alpha hemolysis, a beta hemolysis, or a gamma hemolysis. Now, alpha hemolysis results in a green or brown coloration change, and then the beta hemolysis results in an opaque or transparent result, and then a gamma is no discoloration. And how we perform this is it's determined by streaking. You have a blood agar plate, and I used a 5% blood sheep's agar plate, and you determine it by streaking for isolation. And in a clinical setting, you can also do many stabs of an inoculum into the agar to encourage any anaerobic versions of enzymes to digest blood cells. But after this, you put it in the incubation, and then you take it out 24 hours later, and you're inspecting for the results of the alpha, beta, and gamma, as I was talking about. Then lastly, leads us to the mannitol salt agar test. Now in this, you kind of get a 2-in-1. The purpose of this is you're seeing if your microbe can grow in a high salt containing solution, and if your microbe can use mannitol as a carbon source if it ferments. Okay, there's no reagents used in this. If it's positive for growing in a high salt solution, then you'll see a lot of colonies growth. If it's positive for fermenting, then you'll see a color change from red to yellow. And how you do this test is I did a tube to plate transfer, and the most common type of medium is the phenol red broth, and you put it in the incubation and for 24 hours, and then again you're looking for those results. Am I going to have colonies of growth? Am I going to have a color change of red to yellow? Or am I going to be negative and have neither? So those are the 4 tests that I performed. And I just talked about what the purpose of them are, how you perform them, the reagents used, and then the positive and negative outcomes of each. Now I'm going to talk about my results. So in my results, the first test I did, as stated, was the gram stain. And I first observed a purple cocci. So I immediately knew that I had a gram-positive cocci. I had a gram-positive bacteria. So in making my dichotomous key, this was a great starting place. Like I said earlier, in the Save the Patient activity, we were assigned 1 unknown organism out of 11 or 12. So, at the top of my dichotomous key, I have all the possible unknown organisms I could have had. So under organisms, I have Enterococcus avum, Enterococcus faecalis, Escherichia coli, Klebsiella pneumonia, Ozene, Cocuria rosae, Protus vulgarius, Providencia, Stewart T, Salmonella intrinca, Staphylococcus aureus, Staphylococcus epidermis, Streptococcus sanguineus, and Yersinia pseudotuberculosis. So those I knew at the beginning I was going to have one of those unknown organisms. So in doing the first test, the gram stain, it revealed to me that I had a gram-positive bacteria. So from those 11 in my dichotomous key, I was branched off into a positive or negative gram stain. This first test allowed me to separate those 11 or 12 unknown organisms in half. So now, after my gram stain, I knew I had a positive bacteria that was either Enterococcus avium, Enterococcus faecalis, Coccieria rosae, Staphylococcus aureus, Staphylococcus epidermis, or Streptococcus sanguineus. So the others that were gram stain negative, I'm not worried about them or the next test I would do for them, right? Because I know I've identified that my gram stain is a gram-positive cocci, a gram-positive bacteria. So what led me to do a catalase test, and as well as the blood hemolysis test and the mannitol salt agar test, was that I knew that those 3 tests applied and were extremely relevant to gram-positive bacteria. And I knew this because just of the information that we've learned over the class and different lab experiments and different videos from our professors, and just the knowledge that I've gained. Something else that helped me as well was the identification matrix, which showed the probability of a positive or negative reaction with a certain organism in a test. So the gram stain narrowed it down for me into those that I just listed. And so since I knew a catalase and a blood hemolysis and a mannitol salt agar test were relevant to gram positive, those are the ones that I chose to do next, and that is why. So then I performed my catalase test, and the observations that I saw in my results were no bubbles. And remember, we said no bubbles or a negative result. Then I branched off on my dichotomous key again. So I had a positive and a negative branch for my catalase test, and on the negative I had Enterococcus avium, Enterococcus faecalis, and Streptococcus sanguinis. So now I'm working with half. So I just keep dividing it in half. And that's the purpose of these tests and what I'm trying to do in eventually isolating one organism to figure out what that unknown is. So then from there, I did a blood hemolysis test. And the blood hemolysis test narrowed it down even more. And the results that I saw in the blood hemolysis test was a greenish color, and I saw colonies that were grown. So remember, what does this mean? It means it was an alpha hemolysis as a result. So then I branch off on my dichotomous key, alpha hemolysis, positive or negative. I had a negative result for the Enterococcus faecalis for alpha hemolysis, but positive I had Streptococcus sanguinis and Enterococcus avium. So in this part of my testing, I feel like I'm in a good spot. I have another test to perform, which I chose the mannitol salt agar, and I knew I was getting closer and closer to isolating that unknown organism. And my results for the mannitol salt agar test was that I saw no color change and no bacterial colonies were seen. So remember, what does this mean? This means that it was negative for fermentation and negative for growth in a high concentration of salt solution. So I recorded that, and then that is what led me, in reference to my identification matrix as well, and my dichotomous key, that's what led me to identifying my unknown organism successfully as Streptococcus sanguinis. And the positive result of that was the Enterococcus avium, okay? So I successfully identified my organism, my unknown organism, as Streptococcus sanguinis. And now we're going to talk about, in the discussion part, kind of what went right, what went wrong, any hiccups, what my results mean, and again, just recapping the experiment. So again, we talked about the purpose of why I did this. We talked about the methodology, the different tests, the purpose of them, the reagents used, the possible outcomes, how they're performed. Then I just explained my own results from this experiment. And so now we're going to discuss, okay, what do my results mean? Okay. So going from the top, simply, again, the gram stain, I saw purple cocci. That means I had a gram-positive cocci, led me to know I need to do tests that are applicable to a gram-positive bacteria. I don't need to waste my time on gram-negative tests, okay, that are relevant to gram-negative bacteria. So then I did the catalase, as we talked about. So I was negative for catalase, no bubbles. So what does negative catalase test mean, okay? That mean that there may be anaerobes or facultative anaerobes that only ferment and do not respire using oxygen as a terminal electron acceptor. Okay, so that's what my negative catalase result meant. And then we had the blood hemolysis test, and I told you that my results were alpha hemolysis. So what does alpha hemolysis results mean? And this means that that greenish color that I saw is caused by hydrogen peroxide produced by bacteria oxidating hemoglobin to turn it to that green methemoglobin. So that is what alpha hemolysis means. And then mannitol simply was that was negative for fermentation, which means it was negative for fermentation, and it did not rely on did not rely on that as its sole energy source. And then it was negative for growth in a high salt concentration, which simply means it did not grow in a high salt solution or concentration. So that is what my results mean. And that last test is what gave me the Streptococcus sanguinis. And I did not have any contamination through my experiment. I did have one little hiccup in identifying the mannitol salt agar, the growth in the high salt solution concentration. At first, I had a false positive the first time, and I was just trying to decipher if it was colonies releasing or if it was just the streaking. So I had a false positive for that, but I fixed it. And then I had obviously the negative that led me successfully to the Streptococcus sanguinis. And again, just recapping, discussion based off my results from my test and my dichotomous key, and that led me to isolate my organism and successfully identify it. And again, it was just a really neat experience to apply all that I've learned over the quarter to one project, and it was exciting because I felt like I did save the patient, and just a really good experience, and like I said, something that I will use in the real world. And people are using that right now, today, tomorrow, and yesterday, of what we're learning. So I'm glad I got the opportunity to expand my knowledge and have greater insight moving forward. And thanks for watching. And again, this was my Save the Patient lab final activity for my Microbiology 214.